Hi, I'm Ushma Neill with the Journal of Clinical Investigation, here for another in our Conversations with Giants in Medicine. I'm pleased to be joined today by Cori Bargmann, a remarkable scientist from Rockefeller University who studies the intersection between genes, the environment, and experience. So how are behavioral decisions modified by content and experience? Well, Dr. Bargmann has made this her life's work by exploring the nervous system of the roundworm C. elegans. She's also advancing science and empowering the scientific community and seeking to cure, manage, and prevent all disease by the year 2100 by serving as President of Science for the Chan Zuckerberg Initiative, which is a lofty goal. So thank you for joining me today.
Thank you for inviting me, Ushma.
So I would love to start with a little bit of insight into what you were like as a child.
I grew up in a family with 3 other girls. I'm the third of 4. My parents were immigrants from Europe, and like many immigrants after the Second World War, what they really believed in was education. And so it was a very scholarly household. It was full of books. We all played the piano, we all read, there was art, literature, math. Education was a big part of our lives. And my sisters all went to college, which was actually somewhat unusual in the town that I grew up in. In Athens, Georgia at the time, maybe only about 20% of people went to college after grad school. And one of them is a professor of literature in North Carolina. One of them is a professor of medicine in Virginia. One of them works for the Federal Public Defender in Austin, Texas, and I'm a scientist.
Now, your father is a statistician and mathematician, correct?
Yes, my father was in the Department of Statistics and Computer Science at the University of Georgia. And before that, he had been at IBM working on some of the first early computers at Watson Labs up here in New York.
Did your parents bring their work home? So did your father influence you in the mathematics or even the scientific direction then?
Something I remember very clearly is that whenever we would toast glasses around the dining room table, we had to calculate how many times the glasses were going to clink. So I can still look around a room and think, 6 times 5 divided by 2. So these kinds of games were sort of part of the vocabulary in the house. And part of just understanding that whatever you learned, whether it was about math or about music, would actually be something that would apply in your life and not just something inside the school.
When did you first start to think about science as being something fascinating without it just being an influence from your family?
You know, I grew up in the 1960s during the space era, and if you talk to probably most scientists in that generation, we all wanted to be astronauts when we were little. It was so exciting that men were landing on the moon and capsules were circling the Earth, and it was a time in which science was very celebrated and popular, not just in intellectual communities, but just as part of American life. And so I think it was an environment where that kind of a thought would be in your mind. And at some point, though, I realized that being an astronaut was probably not practical. Possibly the point that I realized that I got sick every time I got on an airplane. So I had to reconsider that thought. And then my older sister Eve went to medical school, and I worshipped my older sister Eve, so I decided I was going to be a doctor too. And I was about 12 years old at the time, so I went and I signed up to be a volunteer candy striper at the hospital so I would learn to be a doctor. And I lasted 2 days in the hospital. It was clearly not the place for me. It was so upsetting to be with people who were sick and not necessarily be able to help them. But something about the idea of medicine was interesting, something about the idea of science had always been interesting. And then not too long after that, I started working in a chemistry lab. And I think the combination of it being very much about learning, but also about doing something practical, about doing something new in the laboratory that someone had not done before was incredibly compelling. So basically, I went into a lab and just stayed.
So there's also a very interesting vignette that I read about you about blowing up a toilet.
Yes, you know, one of the things about science is that it gives you power. Many of the powers that we have now come through scientific understanding that leads to new technologies or to new medicines. And when I was in the 8th grade, in an earth science class, we learned an amazing thing, which was about the chemistry of sodium, and that sodium would explode and burn when it went into water. And my 8th grade friends and I were electrified by this discovery. And so we stole the sodium from the earth science lab and we flushed it down a toilet and blew the toilet off the wall and got into some trouble and had to pay to replace the toilet. But it was a great example of science being a powerful and almost magical thing.
Oh, that's a fantastic story. So everything that you've mentioned so far is math and space and chemistry, so more of the physical sciences, and that working in a hospital was perhaps a little bit upsetting or not right for you. So at what point did you turn towards biology? Was this the college research you were mentioning?
I came into biology in college, and what was happening in biology at the time was the explosion of molecular biology, and in particular, of the ability to clone genes, which was at the time called genetic engineering. I sometimes remember it because now I'm in a place where people do software engineering, a different kind. And the potential that that opened up to understand biological problems in a new way was obvious even to a college student, and the excitement of being able to learn new things using these new approaches was what drew me into genetics and ultimately into a continued career in science.
So you ended up in a bacterial genetics lab in college with the idea then that you were going to go to graduate school for a PhD?
I think by the time that I had been in a lab for a while, I was pretty sure that this was the place for me and this is what I wanted to do. And the funny thing about it is that it's not because it was easy, it's because it was hard. It was because it was really challenging. And if you felt that you got something done, you had really accomplished something. Like the way I remember my entire first summer in the lab as an undergraduate is that every day I would try to do an experiment and I would plate out the bacteria, and then I would go home, and the next morning I would come in and it would have failed every day. And I would go to the vending machine and I would eat some Twinkies. And then I would go back in and I would try to do the experiment again. And there was something about eventually getting that to work that was such a kick and it was such a thrill, and ultimately it was successful.
So at this point you have decided you're going to go do a PhD. How did you decide on MIT, and then how did you decide to go work with Bob Weinberg?
One of the reasons that I went to MIT was that when I was interviewing different places and looking around, it was a place where after the interview I could write down, without thinking, 6 or 7 names of people that I would love to work with. And feeling that there were a set of choices there felt like the right way to make a decision about a place to go. In terms of picking a laboratory, I was just at MIT yesterday, actually, and I saw some of the people that I knew there when I was in graduate school, including Phil Sharp, who had just before I got in there discovered the splicing of RNA that caused introns to be removed so that genes were eventually religated into the mature messenger RNA. And that's kind of the state at which the field was then, right? People were still making just very basic discoveries about what molecules were and how they moved around. I joined the Weinberg lab right at the time that other members of the lab, Luis Parada and Cliff Tabin, were 2 of the leading people doing this, recognized first of all that changes in the RAS genes were associated with the transforming ability of human tumors, and then identifying the exact changes, the molecular changes at amino acid 12 that changed a glycine to other amino acids like valine that occurred in tumor cells that weren't present in normal cells. So that's an incredible discovery. But the thing that I think it's really important to say is that we didn't feel like we were solving a huge problem. We felt like we were graduate students trying to make things work, and most of the time they didn't work, and dropping things on the floor and contaminating our cultures, and just in general trying to figure out the underlying problem of just doing an experiment, and then somehow along the way these other things would happen. I loved being in the Weinberg lab, and it was a really exciting process both at the time to make discoveries and learn new things. And also in retrospect, maybe even more, I feel really proud of what the lab accomplished and even the contributions that I made in terms of moving the field forward. My project in the Weinberg lab was to clone this gene that was associated with rat neuro and glioblastomas. And that was a pretty obscure thing to be working on. Most of the people in the lab were working on human tumors and not rodent tumors, but there were reasons to think that these tumors were interesting. And one of those reasons was that sometimes the immune system attacked them and they spontaneously regressed. And that, we wondered why that was, and ultimately several of us in the lab, including me, were able to identify the gene that was altered in those tumors. And it turned out that the reason that the tumors regressed is that the gene knew and coded a protein that was a transmembrane protein. So it was accessible on the surface of the cell, and it could be recognized by antibodies that would ultimately lead to its destruction. And then after that discovery was made, Genentech put together the knowledge that was altered in human tumors and the fact that antibodies could be used to attack it to develop Herceptin as an antibody to target the class of breast cancer in which this gene is altered. So in the course of about 13 years, I think, the gene was first cloned in 1985, and Herceptin was approved in 1998. You go from, pure science, model system, model organism, through immunology, through medical oncology, through drug development and verification to do something to be able to treat human cancer. And together with Gleevec, it was one of the first rationally designed treatments that actually targeted a mutation that gave rise to a human cancer. And it's a great feeling to realize that basic science works. It's a great feeling to know that really understanding the mechanisms of biology can lead you to the kinds of advances in medicine that we all look forward to.
So I think the question that I have then, especially hearing you talk this passionately about this and having been part of the basic science discovery that led to Herceptin, why did you turn away from that to go to neuroscience in roundworms?
Well, there are several reasons why I changed to doing neuroscience. One reason is that I had always been really interested in neuroscience, and particularly from the perspective of behavior. So I remember my mother, when I was really small, reading to me these books written by Konrad Lorenz, the great neuroethologist, who discovered the nature of and described the nature of instinct in different animals. I still have copies of those books in German, which is how she read them to us. And so I'd been intrigued by that. I was intrigued by the problems of behavior. And what became clear to me after graduate school is that some of these same kinds of approaches, the approaches of molecular biology, might also be applicable to problems in the brain and in the nervous system. And I should say that when I started that was still an idea that was pretty controversial. And there were a lot of people who wrote a lot of papers explaining why there couldn't possibly be genes for behavior and why these things would be much too indirect and have far too many pieces in between to reach an understanding of behavior from a genetic mechanism. Despite the fact that what innate behaviors, instincts, tell you, which is that if there's something that's invariant in every individual in a species, it is in one way or another probably encoded in the genome. But still, how that might work was a real puzzle. And the fact that that was a puzzle is the reason that Mike Young and others won the Nobel Prize in Medicine this year, because they were the first people to really show with great clarity that there were genes whose job was to regulate behaviors, in their case circadian behaviors, and to figure out how they did so, how you could actually have a genetic mechanism for something as complex as an organism's behavior.
So how did you fixate then, after you've chosen your postdoc in the Horowitz lab, still at MIT, that's a C. elegans lab, on chemosensation?
One of the things that I've learned about studying behavior is that you're doing yourself a big favor if you study something that the animal wants to do. That to the extent that you can use an animal in a setting that it finds interesting and engaging and motivating, you are working with the system and you will learn more. And there's a rich history of that in all kinds of areas of biology. So I came to C. elegans because of a couple of different features that made it seem really attractive to me. The first was I was interested in behavior and I was interested in genetics, but it was clear that there was going to be a nervous system between point A and point B. And right at the time that I was deciding about a postdoc, there was this fantastic paper published that described the complete nervous system of C. elegans, all 302 neurons and all 7,000 synapses linking all of them together. And so here was this map of the nervous system that would serve as a way to move back and forth between what molecules did on the one hand and what the organism did on the other hand, because molecules act in and on cells, and cells then in the organism give rise to its various biological properties, including behavior. So this tremendous map was fascinating and seemed like a great foundation. And I should say that that wiring diagram is still the foundation of what goes on in my lab every day. We really do draw on that as a major, major source of understanding.
So you've been described as being a really great worm biologist because you think like a worm.
My postdoctoral advisor, Bob Horvitz, said that it was my greatness as a scientist that I could think like a worm. And worms care about smell. And the reason we studied smell is that the worms really care about smells in their environment. They have a limited perception of light. They have actually quite a good sense of touch, but their main way of interacting with their world is by detecting chemicals that are released by other organisms in their environment. And now, with hindsight, I can tell you that the worm genome has more molecules devoted to detecting chemicals in its environment, smells and tastes, and pheromones, than any other organism whose genome has been sequenced. So it really is intensely interested in this feature of its environment. And the fact that it wanted to respond to these kinds of chemicals, the fact that it wanted to taste and smell its world and make behavioral decisions based on that, was a great reason to use that to study its behavior. It was also, at that point, smell and taste were really fun problems because we didn't know how they worked at all. And so it was a question for the worm, but it was a question more broadly, how you can detect different kinds of odors and tell them apart, and how that information is acquired and processed by the nervous system.
So you established yourself as a fantastic neuroscientist in olfaction and decided to start your own lab at UCSF. So what was that time like?
The fact is, I didn't really know that much about the brain when I started my own lab. And I went to the University of California, San Francisco, and I was hired into the anatomy department of a medical school. And I think that was really brave of them to decide that someone who was working on a very basic system on a strange organism would be a good person to hire. And I think it was also brave of them to hire me to work on the nervous system when, I knew a lot about genes and molecules in worms at that point, but not so much about neurons. And UCSF had a fantastic interdepartmental neuroscience program, and that was the place that I learned about brains and how to think about neural processing and how neural systems work. And I'm just really happy that I had that time. And again, in the way that things are, where you just never know. You're starting out, you're an assistant professor, you're trying to set up your lab, you're trying to figure out how much ice buckets costs. And the assistant professor in the lab next door to mine was this guy who knew a ton about neuroscience, and we were interested in similar things, and we did some projects together, and we worked together, and that was Marc Tessier-Levine, who went on to become one of the great developmental neuroscientists, ultimately the president of Rockefeller University and now the president of Stanford University. And so again, I had this great experience of having a colleague who at the time just seemed as sort of clueless as I was, but was smart and thoughtful and just a great source of stimulation and imagination, and helped my lab advances in its science, and I think our lab helped his lab advance in its science as well.
So you spend a good, 14 years at UCSF, and then transition to the position that you're in right now at Rockefeller.
Being at UCSF was fantastic. It was a great place to learn, but being at a state medical school is controlled chaos all the time. And the medical school is running hospitals, and you're teaching medical students and pharmacy students, and you're merging with Stanford, and you're unmerging with Stanford, and there's managed care going on, and then there's some research going on at the same time, and it's very stimulating. It was a refreshing change at that point to be at a place where the thing that mattered was the thing that I was doing, and the thing that the other faculty members at Rockefeller were doing, which was to really try to move the frontiers of our fields forward through our research. And the great thing about Rockefeller is that it has this single-minded focus, and it's small, but it's embedded in a community with other institutions literally right across the street. So you are at Memorial Sloan Kettering Cancer Center, and across the other street is the Weill Cornell Medical School, and some of my favorite neuroscience colleagues are at the Weill Cornell Medical School. And some of my favorite developmental biology colleagues are at Memorial Sloan Kettering. And so at Rockefeller, and in that little corner of New York, we get the best of both worlds. We get to have our own place with our own identity.
In hearing you talk about all of this and your passion for science and the way that you're so enthusiastic about lab work and these discoveries that you've come through, it's interesting that you started taking on some slightly more administrative roles, like when you were the co-chair the Obama-initiated BRAIN initiative. Can you tell us a little bit more about what that was like and why you did it and what you set it on the road towards accomplishing?
Why Francis Collins asked me to do the BRAIN initiative, you will have to ask him. But I was very excited by the fact that here the President and Congress had allocated money for science, for new programs in neuroscience inside the NIH. And the NIH had chosen to do what I just said, to say, let's go to the scientists, let's go to the neuroscientists and have them decide what this field really needs to move it forward. That they were willing to trust that rather than saying, well, we're at the NIH, we know what that is. And Francis Collins had experienced that himself in that the Human Genome Project was set up again. It was the scientists who were the experts in the field who got together and said, we really think it's time to sequence the human genome. And that idea bubbled up into the National Institutes of Health as the way that the science moved forward. So I felt that this was an opportunity to do the same kind of thing of helping a community take the next steps that would take more thought than the next grant or the next small program.
Well, so then furthermore, you've taken on an even bigger role as president of science for the Chan Zuckerberg Initiative. And as I mentioned before, the tagline that I've read is that it's to cure, manage, and prevent all disease in this century.
So Mark Zuckerberg and Priscilla Chan decided on the birth of their first child that they were going to give away the vast majority of their wealth to try to make the world that their children would live in better. And Priscilla is a pediatrician, and she has been very motivated in her own life by the fact that the children that she could not help were the children for whom there was no help. And the knowledge that there were a lot of diseases that we just don't know how to cure or prevent or treat or manage now. And so this was the inspiration for supporting science as a way to move the medicine forward. It's an incredibly generous gift, and it's an incredibly great opportunity to be involved in this. Now, curing, preventing, and managing all diseases is a strong statement, but the end of the century is a long way away. And the goal is to support science and technology that will make it possible to cure, prevent, or manage all diseases by the end of the century. There are a lot of people working toward that goal. The Chan Zuckerberg Initiative is not going to be the one group that solves all diseases, But what we can do is think about where we can help out and where we might be able to make a differentiated impact to move things forward. I am a biomedical scientist. I believe in science. I believe in the progress that science has made toward medicine in the past, and I believe that it will make that progress in the future. And there are a lot of great scientists out there, and the question is, how can we accelerate the progress that they make? How can we make science better? And coming from Silicon Valley, which is where Mark Zuckerberg is, there are some perspectives about how to make systems more efficient, how to make them move faster. And they involve things like software engineering and data science and rapid transmission of information between different groups so that everyone becomes smarter faster.
So why start with the Human Cell Atlas?
I love the Human Cell Atlas project as a first project for the Chan Zuckerberg Initiative because I think it demonstrates what we're trying to do in a way that should be easy for people to understand and help, and also then a way that people can help us figure out the next things that we should do. So, every human disease has a cellular basis. Every human disease represents some of the cells of the body either doing something they're not supposed to do or not doing something they're supposed to do. There are 37 trillion cells in the human body. We don't actually know how many different kinds of cells there are. And almost every time we learn more about the cells in the body, we actually learn something that's medically useful. So what is the cell atlas? There are 37 trillion cells. You want to know like how many kinds are there and how many of each and where are they and who are their neighbors and what are their molecular compositions. And that's possible now because of advances that have happened in a variety of fields of single cell biology, where you can actually now look at a single cell and say what RNAs are in it, and often many proteins, and look at its location in a tissue slice. It's a big advance, a big jump in classical histology that's happened just in the past few years. So this is possible, but it's a big problem. It's like the Human Genome Project, except that there are only 25,000 humans genes, and there are 37 trillion human cells. So it's going to take a lot more work. The idea of the Human Cell Atlas came from the scientific community. It bubbled up from the experts in the field, just like the Human Genome Project did. These people had sort of self-organized and started writing white papers about what they thought was possible. And they're the scientific leaders, but there are a set of things that are going to need to happen for that to be successful. One of them is that this technology development, so that everyone can sort of develop shared tools and shared technologies. And so at the Chan Zuckerberg Initiative, the first request for grant applications we funded was to develop better tools and practices so that there would be common standards in the field. Another big problem turns out to be this is going to be an enormous amount of data, much, much bigger than the existing databases. It's going to include all the kinds of data you get out of genomics and lots of data about images and ultimately other kinds of information. So because at the Chan Zuckerberg Initiative we've hired software engineers and computational biologists, we can actually help people in the academic community build a big data platform that is in a modern software architecture and that is suitable for doing this kind of large-scale data sharing and analysis. It's going to be a cloud-based platform instead of the sort of physically based platforms that typically exist in biology. So here, it's very literally people from the commercial sector are working with the great experts in this area in the academic sector to build something together that's better than either of them would build alone. So that's something that we feel we can push forward. And then finally, there's just a lot of really interesting computational and data and analytical questions, and we're doing that partly through our own in-house computational biologists and partly by funding external people in computational biology. So it's pulling together this idea of raising all boats by developing a better technology, of using the things that have developed really well, but not yet been fully incorporated into biological systems, like data platforms and cloud-based data architecture. And finally, the third part that we're really proud of is that we are trying as much as possible to make this a fully open resource that everyone can use and everyone can share. And John Sulston, who started his life as a C. elegans researcher, then became one of the leaders of the Human Genome Project. And as a leader of the Human Genome Project, he he passionately argued for and ultimately was able to convince people that the whole human genome should be completely open, that everyone should have access, that all the data should be made freely available right away. And that had been a philosophy that had grown up in the elegans community, and that's the philosophy we're taking to the Human Cell Atlas, that everyone should have access to this knowledge about the human body as a foundation for subsequently studying all kinds of questions in health and disease.
If you were not a scientist, what else do you think would have captivated you?
I love being a scientist. The only thing I can come up with is being another kind of scientist.
Well, what other field of science then do you think would have held your interest?
I'm just going to give you some bad answers. I love neuroscience. What could be a more fascinating problem than understanding the human brain?
Okay, well then you're committed. Well, it has been a pleasure hearing a little bit about the things that have motivated you. So thank you so much for joining me today.
Thank you, Ushma.